Okay, so today we are going to cover the BioFlux system. It's a specialized platform which allows you to perform shear flow assay under physiological condition. And we will have a focus on microbiology applications. So going back to the origin of this type of technology, the term and the concept comes from the original flow cell, which is utilized in laboratory already from decades. The, let's say, issues and the obstacles of this classical flow cell are the amount of samples that is needed to be utilized, of course, the throughput, which is quite low, and also the risk of contamination for the sample and for the operator itself. So with BioFlux, we want to overcome this issue with an innovative type of platform, which is composed by three main components. So this is the controller, which is providing the pneumatic flow, the so-called shear flow. It controls the environment and the temperature. The interface, which is directly connected to the tubing. Each of these tubing is going straight into the plate, controlling different region with different flow velocity. And the plate itself. So the plate itself, which is composed by different type of format, we will discuss later on the type of plates, but the concept is that we apply this interface on the top of the plate, we seal it firmly, and we apply some pneumatic pressure inside the plate. Inside the plate, there are different type of microfluidic channels, and there are two different input and output. So the sample is injected in here, the pneumatic interface is applied, the controller is deciding which kind of shear flow we want to input into the channels. And then after sort of recirculation of cells or bacteria here, we reach the observation area. So this is the observation area. The plate are having a glass cover slip, so the user can easily get some very nice quality images, either confocal or conventional microscopy. So this is the main concept of the 48 well plate, which has one input, the serpentine, the observation area, and the output. So the concept is quite straightforward, but definitely allows the user to keep all the biological material inside the plate within the two layers that are composing our special plate. A few microliter of biological samples are needed, not milliliters like in the classical flow cell. And you can definitely multiplex quite a lot your assay, because using a 48 well plate, you can have up to 24 parallel assays. So you are sharing two wells for each assay, resulting in 24 parallel different assay. And you can see here, once again, the schematic representation. So you can imagine the example with cells. This is the inlet. Here you input your, let's say, 10 microliter. The flow starts, the sample it's recirculating here in this observation area in the serpentine, and directly underneath you can use any inverted microscope compatible with brightfield or with fluorescence, can be utilized with the BioFlux plates. As you can see here, the software can control the different regions independently. So if the user wants to keep channels on or off, there are 8 different independent input and output. The user can even reverse the flow from output to input if they want to change the behavior, the adhesion of the cells or the bacteria. And this is the example of the 48 well plate, while if we are going to check another type of concept, another type of format is the 24 well plate, where the throughput is slightly decreased, but we have higher flexibility from the point of view of the experimental design. So the 24 well plate comes with 2 different input, as you can see here. So A1, B1 are the 2 input and 1 output. Such a situation can be convenient if you want to, for example, have some different dosing of some compound. So here you input your biological material, your cells or bacteria. Here you position your treatment, and then you activate this channel, for example, only after certain hours that the cells are recirculating into the channel. So this is increasing the flexibility, and the user can even create a flow gradient in a way that if you want to check the behavior of some anaerobic bacteria, so you want to change the oxygen percentage, you can have two pre-mixed gases coming from input 1 and input 2. So here can be, for example, some input with classical 5% CO2, or here can be with different concentration of oxygen. And this will allow you to create a flow gradient inside the channel and will change the behavior of your cells. So the throughput of this plate in this case is slightly different. We have, as you can see here, 6 parallel experiment with the 24 wells. How BioFlux works, it's a very precise platform. As I said, the interface is applied, the pneumatic pressure is applied into the input well, and the serpentine receives the fluidics observation area, and then the fluidics come straight to the output. So you can imagine that in between here, there is actually the serpentine and the viewing area, which we just checked. The important point of this technology is the unique accuracy and precision in changing in range of really millisecond, microsecond, the flow instantly. It's not volumetric based, it's pneumatic based. So we can really quickly stop, increase, decrease, reverse the flow, and this can allow the accuracy into the results as well. Here you can see the example, the responsiveness of the instrument, it's really impressive. And so we can, you see, reverse and push again forward the flow. This is some example of monocytes flowing. And it's instantly stopped and restarted due to this difference in pneumatic flow compared to the classical volumetric one. The dynamic range is quite high, so the user can decide to apply a very, very low stress, which means 0.1 dyne per centimeter square up to 200 dyne per centimeter square. And this allows the possibility of deciding which kind of, let's say, shear flow you want to apply depending by the goal of your experiment. So for example, if you really want to stress the adhesion of some bacteria, you want to go maybe up to 50, 100 or 200 dyne per centimeter square. If you simply want to flow some cells inside, you can lower down the dyne per centimeter square to 5 or 10. And the precision and the accuracy is not only in connection to the pneumatic flow, but also related to the order of this flow of fluidics in parallel, which are running into the channels. So here we also checked, based on the principle of the fluid dynamic, that we are really able to obtain smooth order flow in the parallel layers that can guarantee that in all this area of observation we can see the same behavior of the flowing of the fluidics, and this is quite important. So the plates are composed by some special composition. So here you can see the well plate. The upper part is actually the things that you see as a wells. So it's a classical plastic as you can find in your conventional microtiter plates. In between the well plates and the cover slip, there are the PDMS containing the microfluidics, which I was showing you. And finally, we have a nice glass cover slip on the very bottom of the plate, and this allows you to get quite nice quality observation of your high resolution of your images. Here you see also the characteristic of the plate, technical specification of the 48 well plate. So very high throughput, 24 assay in parallel, from low up to high shear flow, up to 200 dyne per centimeter square. So usually the microbiologists are adopting this possibility if they want, for example, to apply a very strong stress to their biofilm adhesion, or if the users are interested as well to try some dual flow chamber, so in a way that you can create a gradient of gases, they also use for other experiment the 24 well. So there will be two different interface, one for the 48 well plate, one for the 24 well, and you can get quite nice flexibility. The plates are delivered sterile. As I said, few microliter of samples are needed, and everything which could be infective for the user, it's really contained inside this layer. So it's really totally safe. This is the unique advantages. Once again, other instruments are using some flow chamber, some channels around the laboratory environment. They are using some kind of syringe volumetric system for pumping the flow. So the accuracy, it's not comparable. The amount of sample needed, it's really high. And of course, based on this characteristic, you cannot get the same throughput and same precision as the BioFlux. The duration of the assay, it's also related to the plate type that you want to utilize. Obviously, if we are going to apply higher shear flow, we have less duration because the fluidics by higher flow, higher stress is drying up faster, and it's ending in the output wells faster compared if we run, for example, at low dime per centimeter square. But each plate has also different channel characteristic in terms of channel diameter, in terms of channel length, and of course, as a number of experiments. So here you can see 48 well plate, up to 24 parallel experiment, 24 well 8, and the 6 well plate 3. So we have a lot of flexibility. For example, in the 6 well plate, you can put up to 70 milliliter. In the 48 well, 1 milliliter, and the 24, 3.5. The duration of the experiment can be very long for assay, which doesn't require really long stress. With strong stress, I mean, so you can see up to days. Or if you want to apply some stronger stress, like here, usually these experiments are not lasting more than half a day. There is also possibility even to increase the multiplexing using this so-called quattro system. So this is like, it means 4 plates in parallel connected to the same controller. And this is something adopted, for example, by pharma company, which are working into toothpaste production. They are very interested in oral biofilm, and they want to multiplex their assay for the drug screening. So the components, as I mentioned previously, are the controller, the plates itself, and the software, which is actually deciding the settings of the experiment. So the user using the controller can decide the shear flow, can turn on and off specific channel and check the temperature. The plates, we already covered it, excuse me, we already covered it briefly. And the software, which is actually our setup interface, which allows the user to choose the different conditions. There is even more advanced configuration which we are able to provide for user which they don't have any inverted microscope in their lab. We have one example installed in Wrocław, where they are working a lot on biofilm, and they decided to have basically a ready-to-use solution with a really nice, high-quality inverted microscope from Zeiss, fully automated stage, X, Y, Z, and ready to use also software interface, which Fluxion has optimized for specific analysis. For example, we have software module for analysis for microbiologists, for immunologists, for hematologists, depend by the field of interest. And here you can see the example of the control software. So let's go through like a quick simulation of how to set up an experiment. Here you can see the 48 well plate. So the user first has to decide what kind of plate you have. And this is really easy because when you log in into the software, you just input the catalog number that you see on your plate. So the software automatically will choose the right plate. The fluidics, it's quite important because of course there is a different viscosity, different density of the fluids. So the system is also estimating the whole characteristic of the flow, the microliter per minute, the duration of the experiment according to the viscosity of the fluidics. So here you can see, for example, from water up to whole blood, there are different setup. And then the user has to decide the shear flow. So the shear flow, as soon as you input, for example, your assay at 2 dime per centimeter square, there is an automatic calculation of the subconsequence flow in microliter per hour, the estimated pressure in PSI, and also the runtime. So it means that if you apply this type of fluidics, if you have this specific volume inside, because you also inform the software about the volume. It will take about 5 hours to complete the experiment. Additionally, when you are acquiring your images, because the idea is that, of course, the microscope will acquire some time-lapse images of the observation area directly inside the channels, whether it will be just phase, brightfield, or fluorescence imaging. You can process directly this. If you have the BioFlux 1000 Z, which is the full configuration, you can process this directly with our optimized software module. For example, you work with bacteria, you can use some dyes to check the adhesion or the viability. Or if you work with cells adhesion, for example, you can even check the velocity, the intensity in connection to some specific adhesion molecules. Going through the application, so we have covered the technology. The application, the main use of this technology is for, as I said, immunologists, hematologists, and especially microbiologists. Let's say 50% of our users are definitely microbiologists interested in the behavior of colony of bacteria under this special condition of shear flow compared to the classical static environment. But of course, I don't exclude the possibility to use the system for even more classical assay, just done in a different way than the conventional microplate. So, for example, there could be wound healing done using the flow, or the chemotaxis, the invasion of the cells by coating with some matrigel the channels. So the system, it's really open for new ideas, new application according to the user wishes. The importance of the biofilm, which is our main topic of today's call. So biofilm are seen every day, whether it's a oral biofilm, or we can find this as a main, let's say, starting point of most of the infectious diseases which are seen in Europe and in US. And there is a lot of interest in that. There is a lot of interest in this type of field, because you can see here that during the years there is an increase of billions invested to study the behavior of the bacteria, because in the hospital, in the medical field, they found a correlation, a strict correlation of 80% of all infectious diseases have a really biofilm start, with biofilm components. What is the importance of using the BioFlux for this type of assay? So the classical assay, which is done on a static environment, definitely is a quite simple setup. It's easy to load your bacteria into the well. You can do high throughput, and it's not really expensive. But on the other hand, you are missing a lot of information from the point of view of imaging, because you cannot see nicely the single layer of the bacteria. There is not really interesting morphology data for adhesion, as well as the real physiological condition where the bacteria are behaving are not in a single well plate, are not in a static environment. But if we want to simulate, for example, the blood flow, BioFlux definitely brings the accuracy of the experiment to the 100%. So the simulation of the microenvironment where the biofilm and the bacteria are proliferating. And here you can see that with BioFlux, we bring, from the point of view of throughput, high throughput, because we can do up to 24 parallel experiments, but also very good and high physiological relevance, because we work in a simulation of the environment where this biofilm is created. So also the flow cells brings you some physiological relevance, as I show you in the beginning, but definitely it's more complex, more risky for some pathogenic bacteria, as well as very low throughput. So what is the idea of the BioFlux? There are different approaches. There are many publications I can share with you in the end of this call. There are different models, in fact, utilizes on this. So the simple proliferation of bacteria and the interaction within the colony of bacteria to create biofilm, as well as I mentioned, using the 24 well plate, we can try also to change the gaseous condition to create this dual flow gradient of gas to induce, as you can see here, to induce the different oxygen condition in one part of the channel, while here, for example, you can have 1% O2, in the other part 5% O2, and this can be interesting to compare the behavior of bacteria and the capacity of proliferation and the strength of the biofilm in this part of the channel versus this other part of the channel. And finally, there is also another approach, which is definitely the evaluation of the host-pathogen interactions. So there can be some nice, there are a lot of nice example of cell-bacteria interaction in these conditions. Here you can see one, let's say, way how to approach this biofilm and this bacteria proliferation. So we are using the classical BacLight, so the light dead dye for bacteria, and we have tried here our negative control just with water, with second condition positive control with 1 ppm of hyperchloride, and going further, increasing by 10 times, we can see that there is an increase of killing of bacteria. So this can be a quite interesting, easy example of where to start in this field. Or simply the different behavior, the different morphology change. Here we can see some Pseudomonas fluorescens, where after applying the stream for 29 hours at 10 dyne per centimeter square, there is some certain morphology and some certain aggregation, while after 50 hours, we see completely different confluency of this bacterial layer. And then we can actually understand a bit more how these specific bacteria are behaving under longer time shear flow, or comparing the mutant with the wild type. So this is Staphylococcus wild type under 10 dyne per centimeter square as some certain way of aggregating versus, for example, the mutant, which shows one more uniform monolayer under the same condition. Yeah. So there are many ways how to approach this field, and definitely microbiologists can find a lot of advantages in using this type of technologies. Another nice example is the way how these same bacteria are actually expressing gene in a different way when they are under shear flow, and they are, let's say, creating this collaborative environment. So they are adhering very strongly, they are increasing their biofilm, you can see, and the gene expression is increasing after the adhesion and the strengthening of the biofilm. So this is also another quite interesting model to do on at high shear flow. In this case, I think it was applied 20 dyne per centimeter square. So increasing the adhesion, increasing the cooperative behavior within the bacteria, and creation of the biofilm results in higher gene expression. As I mentioned, the BioFlux allows the user to go with very high resolution, high quality analysis. We have a 170 micron cover slip, glass cover slip on the bottom of the plate, which allows the user to do more advanced processing of the images obtained. For example, we can even do confocal images out of the bacteria, as well as we can do some nice post-processing like this sort of 3D holographic imaging if we want to get the real information, for example, of the volume created by the biofilm over time, and not only the area, the morphology, or the fluorescence signal coming from this specific dyes that we utilized. So here you can see the biomass, cubic micrometer, the thickness, the roughness. So there are really a lot of possibility to go into very deep and detailed post-processing. And here you can see another example of co-culture. That's another interesting model to take in consideration. So Pseudomonas with E. coli as a cooperative behavior, that they are creating actually stronger biofilm by increasing the flow rate. So this is also quite nice simulation of what could happen in the real environment, in the real infective process. And other type of bacteria, other type of bacillus. So here we have placed some cell tack, which is kind of adhesive coating matrix. And also in this case, this adhesive matrix would kind of simulate the epithelium of our blood vessels. So we have noticed that the Bacillus subtilis was in fact increasing the motility and the adhesion after increasing the flow rate. So there is need to have flexibility into this shear flow from the point of view that we want to study in the same time, for example, for 2 or 3 bacteria in the same well, we want to have different condition and different replicates to better understand the behavior mechanisms. The third model, another way how to approach this is the host-pathogen interaction. And here you can see the cytopathic effect of the Pseudomonas aeruginosa. So here we see the really important difference of the behavior in the image. Number A, when we start the flow 1.5 hour after and 3 hours after the flow. So we see really in a 3D way how this Pseudomonas was capable of having interaction with cells and capable of infecting actively this layer of epithelial cells, even after quite a long time of high shear flow. Why not to take in consideration other type of microorganisms? So in this case, we can work also with yeast. Until now, we discussed about bacteria, but the BioFlux is opening doors to many type of microorganism related applications. So we can study also the capacity of yeast to create the biofilms. And here you can see a quite nice example of fluorescent images of yeast co-culture together expressing 2 different markers. So here also you can notice over time at lower shear flow the cooperative behavior of these 2 co-culture to create a solid, strong biofilm over time. And another example here with some HUVEC cells with yeast. So also here another host-pathogen interaction inside the channel where we wanted to check the infective capacity of and the adhesion capacity of the yeast itself into the layer, the monolayer of the endothelial cells. So to conclude this brief introduction into BioFlux for microbiologists, I really strongly believe, and we saw it also based on our direct experience, that the BioFlux allows the microbiologists to really understand the whole story, the mechanism of not only the way how the biofilm is created, if the bacteria are able to create the biofilm, but the adhesion steps, the aggregation, the creation of the microcolony, all the way how these bacteria are cooperating and proliferating in the creation of the biofilm, as well as this can lead to a better understanding how to disrupt this infection, how to disrupt this aggregation of biofilm. And this is an instrument which is really a lot utilized by a lot of military hospitals and a lot of, I mentioned pharma company connected to, for example, oral biofilm. They can find a lot of benefits in using this technology. So to summarize, we can see different aspects of monitoring biofilm, so the growth itself, the adhesion, response to compound, but also a bit more advanced host-pathogen interaction. So we have a really nice overview to make us understand much more compared to the classical microplates where we work and we operate in a static environment which is not simulating the real life conditions. There are hundreds of publication for microbiology in really important journals like Microbiological Methods connected to pathogens like the Aureus, E. coli, so the impact of different treatment, the impact of different antibodies, classical viability screening. I will be really happy to share with you all the protocols and the application note and reference that we have in microbiology, because as I said, this instrument is utilized by 50% of our users are microbiologists, and here you can see some nice example of universities which are adopting this this instrument at this very moment. And of course, I need to mention also our local user. We have proudly 3 installations in Poland, at the University of Krakow, one at the Children's Hospital in Krakow, and also the third one at the Medical University in Wrocław, where they are really specifically focused on the biofilm. So I will be happy even to share with you a reference. Talking about briefly another type of application which is not directly connected to the topic, today's topic. Here you can see which possibilities you have, how complex you can bring on your assay, not only with the classical host-pathogen interaction. So this is a nice model done by users in Krakow, which they are interested in simulating a more complex situation, which is really happening into the blood vessels. So they, first of all, input into the channel endothelial cells, which are creating actually the monolayer, so the endothelium itself, and then they afterwards input into the flow red cells and platelets, and they have tried to apply some kind of irradiation to see how basically this endothelium was disrupted by irradiation, and how the platelets were able to recover that. So it's really impressive, this kind of method. It's really complex and can bring really new insights compared to the classical methodologies. So once again, here it's some of our customers. We have 3 installations around the Eastern Europe, and we are waiting for more. And at this very moment, I would like to thank you for your attention. I will be happy for any comments, any questions. This webinar is going to be recorded, and we will share it with you.